Curious about antiviral development, antiviral drug developments. I'm a former HIV researcher and now program officer in AIDS, and we've had immense success. It took maybe 10 years from the discovery of the virus to this first two classes of to bend the curve essentially.
Right.
What's the hang up for other things like flu or other viruses? Is it basic science? Is it translation? Is it industry's not interested?
Well, I think so. Well, first of all, the person that you're sitting, Mindy Davis, that you're sitting at the table right here, introduce yourself to her, and you guys can talk about antiviral drug discovery because Mindy's got a lot of that in her portfolio at DMID. So what is the hang up? Well, I think it depends upon the virus. I think people right now for herpes simplex virus, for example, think that we have some pretty good antivirals, and we'd really like to have a vaccine that was effective, particularly genital herpes is both HSV2 and 1. But for that specific virus, I don't know that we're investing a lot of money in new antivirals, unless something that looked spectacularly promising came along. But I don't know what safe and effective, we already have that. For Zika, we would like to have something that, if it could, prevent a pregnant woman from becoming infected if they were living in an area where an epidemic was occurring, but that was still safe. And also prevent shedding, because we know in the case of Zika that it is shed in seminal secretions for a long period of time after, perhaps a long period of time after infection is over, and the initial infection establishes itself and there's no virus systemically circulating. So that's a problem for women that might be getting pregnant. I mean, if the worst consequence is congenital Zika syndrome. So, we have all these new viruses and we have efforts to develop, to identify druggable targets and identify compounds that would be effective. But I think it depends upon the virus that you're talking about, where the biggest obstacles in development lie. In the case of flu, which I think I heard you mention, it's hard. Well, we have a few things in development. There are monoclonal antibodies, too, that people believe are broadly neutralizing and might be effective therapeutically in someone with symptomatic influenza. The standard for testing from the FDA right now is a comparison to oseltamivir. So there's problems in conducting that trial because oseltamivir is licensed for, give it within the first 48 hours of symptoms. So, it's hard to recruit those patients. Most people that get sick that get influenza infection don't have very much in the way of symptoms and don't necessarily seek medical care. And if they do, it might be outside of that window. Or by the time that you would enroll them in the trial and they would actually get the product into them, it would be outside of that window. And the standard for licensure is non-inferiority to oseltamivir. So, it's maybe in that particular case, if you really had promising compounds, you would work with the FDA to change, come up with a reasonable standard for testing a compound, because we do need alternatives to oseltamivir. We know that.
But I think that it's a great example in HIV. We figured out very early that single drug regimens result in resistance. Tamiflu resistance occurs within a month of rolling out the Tamiflu. So why not having two drugs, two targets, right? And is that a priority? Is that prioritized?
They have done drug trials with combinations. The one advantage, just to go to your HIV example, HIV had a very good surrogate marker, and licensure of a new drug could be done based upon durable virologic suppression, right? So in influenza, viral shedding hasn't always compared with good therapeutic outcome. That's a tough one. There's no real viremia to speak of. So the best surrogate marker, and especially when you're looking with such a short window of opportunity for therapeutic intervention, that was a hard one. And similar quandaries arise in the case of Zika, because could we license a vaccine based upon just eliminating circulating virus, viremia? And since so many people are asymptomatic, that wouldn't fit the FDA's usual approach to drug licensure. Usually it has to show clinical benefit. The reason in HIV that you could make the strong inference of clinical benefit was because you had that proven durable virologic suppression was a proven indicator of long-term clinical benefit. So they started accepting that as an endpoint in clinical trials and licensing drugs based upon that. So, you know, basic research and clinical research that helps point to good surrogate indicators that could be used in licensure applications and hold up at the FDA, which is always going to look for that sort of evidence. That's another challenge for researchers, particularly not only in viruses and everything. Yes.
Curious, talking about Zika too, like when you're sort of doling out the extramural funding, like Zika's been around for a long time in Africa, just hanging out, not really anybody's concern. Yeah, it's Zika. Came to South America, yeah, it's Zika, and then all of a sudden all these microcephalies, and it's like, holy cow, where did this come from? Why is it doing this now? How do you, is there a way to fund the research on the virus, the bacterium, the pathogen that we know does this here, but what if it came here and changed? Like, how do you...
Or, like, pick the ones that are going to cause terrible impact. They look like they're just innocuous.
Right, yeah. How do you even...
Yeah, that's a good question. I think the approach that we've taken is that we haven't fully developed the platforms for this, but viral families that are known to be threats to humans, we are trying to have sort of a prototype approach that we work through all the steps, or at least we know we can work through all the steps quickly for vaccine development. You know, Zika is an arbovirus. It circulates with the same vector in the same places that dengue does. So we haven't proven yet that we don't actually have a great dengue vaccine. If you read the news, you know what happens to Sanofi when they put theirs on the market. But the idea that certain families, there could be certain successful approaches for developing a vaccine, and then if something new of that family all of a sudden emerged and could cause something terrible like Zika, you would have that platform that you could use and you could move more quickly than if you were just starting from scratch. So that's our current way that we think we're thinking strategically, and hopefully, you know, people are getting faster and faster with rapid platform development. So that's a good thing, particularly for antibodies, but then the next step would be for vaccines, and maybe even for vector vaccines. So it's a challenge. What you just described was a challenge because no one was expecting that. Nobody even knew what it was until all of a sudden. Yes.
I'm thinking along the same lines as the previous question. It seems like CDC is historically has been your surveillance organization, although NIH and NIAID support some surveillance, but not a lot, it seems to me. And surveillance is what's going to show you what's coming up next, what's coming out of Africa or out of North Carolina, wherever. Is there any interest in sort of increasing our portfolios of human and animal surveillance?
At the NIAID, I think that we want to work closely with the CDC because I think surveillance is part of their mission. And we supply viral isolates to them all the time for very, you know, when that's an interchange that they need for asking some question that they need to ask. But they probably choose pathogens to focus on that they know cause disease. And in the case of Zika, before it caused disease, we didn't know that it would cause disease, right? So in answer to your question about surveillance, I mean, I think we want to collaborate with the people. We want to do our part to make surveillance effective by collaborating with CDC or WHO or any partner that has that need. To do it ourselves, we'd probably be running into overlap or even destructive things with the people that we should be collaborating with. So I think not going into that wholesale would be my answer to you. I mean, we're primarily a research agency. And there are, there's tons of viruses out there that are never going to cause problems for humans too. So that's the other thing. Do we want surveillance in all those? Maybe we do if it was just one mutation away from something bad. And that's a research project, though. All right.
I've got a couple of questions, if you don't mind.
Sure.
Could you just take a moment and talk about your personal journey as to how you got to NIAID? And the other piece of that is, I know we spoke very, very briefly on the phone about it, but could you talk about being a woman in science? It seems to be there's a lot going on lately about women in science, the Me Too movement, all that stuff. Could you speak a little bit about that as well, please?
Yeah, I don't have a Me Too story to tell. I mean, it's probably
Good, that's good.
So my personal journey, as you heard my bio, I trained in infectious diseases. I was at Hopkins for a long time. I was doing clinical work in HIV and sexually transmitted infection. So it was very public health oriented. I mean, public health practice oriented. And also, I saw infectious disease consults on the wards. I was general medicine attending on the wards. And I liked all that. And I don't know that I had a plan to move. I was just looking to see what else I could do. And then I got this, I think, an email from the head of the search committee for the deputy director of the Division of AIDS, and I thought, oh, this could be interesting. This could be something to do. And I will say, the one thing I will say at Hopkins, and I know that there are people in the room who recently left Hopkins, so they can probably sympathize with this part of the job. I mean, everything that, you're responsible for bringing your money, your full salary in. There's nothing that is given to you. There's nothing that is, you don't get like the 25% that you can just sit around and develop and think and do whatever you want with. It's every penny you have to bring in yourself. So I had contracts that brought in my salary for director of the sexually transmitted HIV program at the health department. They followed the revenue that I brought in for doing clinical consults and for being in clinic a half day a week, and every little bits and pieces of everybody's grant. But it was just, it just kind of wears on you when you have to do that, and you have to pay people. I mean, you have to bring in the money for the people who work for you too, and that's where it gets kind of dicey because it's all soft money at end. Funding periods end, and everybody that thought that they owned a piece of me was never totally satisfied with how much they were getting out of me. They sort of sometimes wanted, if they're getting 50% of your salary, they want everything that they want totally when they want it. So that was getting a little bit old. So I was looking for what else I could do, although I loved being at Hopkins. It was fun. I mean, I liked my colleagues and I liked the work and I liked being an infectious disease physician. So when this other opportunity came up, I just thought, oh, I'll just look into this. And you sort of assume when you see ads that, and sometimes this is true for NIH, there's a acting that's the incumbent, and they just have to, for Title 42 positions, for leadership positions, they have to advertise and interview a certain number of people. But in this particular case, when I talked to people who worked at NIAID, they were like, no, they really, they've gone through those ropes, and they want a person from outside, they want fresh ideas, and so I did the interview, and it seemed like, as I did the second interview and met with Tony, it seemed like something that was really interesting and new, and I didn't have to worry about my pennies of my salary making it all add up. So I guess that was what made me change that at that juncture. So that was 8 years ago. And then coming to be, well, maybe it's not such a dramatic change to go from deputy director of a big division to director of the big division. So I sort of always had my eyes on what was going on in DMID because we collaborated with them. I talked to a few people when I heard that a retirement was creating this new opening. And so, I threw my hat in the ring for that too. And this is where I got with that. So that's my journey professionally. So your other question was being a woman in science. I was going to medical school when women, we weren't much of a minority. I was probably like not more than 50% of the class were women, but it was starting to approach that. So numerically, I've always been surrounded by plenty of female colleagues in residency and in infectious disease fellowship, and even as an attending at Hopkins. It seemed as though at a certain point, women started to find the challenges of the demands of being a professional in that environment and having more work to do raising children, and not necessarily equity. And I mean, physiologically, you can't really, you gotta get pregnant. I mean, you're the one that gets pregnant and has to deliver, so there's no equity there. But then also just organizing a household and doing the things that you do in America these days when you have kids. You chauffeur them around, you go to swim meets, you go to soccer practice, and it always seemed like women, my colleagues with kids, were always doing more. They were always the one that was worried about the daycare deadline, that they had to, get there by 6 o'clock, they start charging you $1,000 a minute or something like that. And so that inequity between men and women, that's when it starts to hurt people's professional development, and the women sort of drop out or are unsuccessful compared to male colleagues who might not be married to women who are trying to juggle all that stuff. That was what I noticed, and I don't know what the solution. I mean, I think the solution there is for workplaces to just be more flexible when they can be, and maybe we need much better childcare for everybody, more available, maybe on site. I don't know. I mean, that's where I think inequity comes in. And now here at the NIH, I think one of the inequities is certainly men stay around for a real, you can be a really effective scientist into your 70s, or maybe even there are people around here in their 80s that are still in leadership positions, to some degree of leadership positions. And so they just reflect who was in training and who was successful 40 years ago, right? So that's why I think we don't have as many women at the top in any of our institutes as we have men. I think that's my reflection, but it might not be. But certainly at the GS 14, 15 level, we have women, and I don't know how that compares to 40 years ago. I imagine it's better now, more equity now. I don't know if they feel like they're treated equally. I hope they do. See that?
Is there any move within NIAID to promote diversity in leadership? Because, I mean, what was striking about the org chart was that you're the only one that doesn't look like everybody else.
The only one that's, well, they used to say that all the people in leadership are men with white hair. And then there's one division director that doesn't have, that's male but doesn't have white hair. So, but yeah, everybody else is a gray-haired man. And I think there's no mandatory retirement age. So people serve until they want to retire. So unless they leave for another opportunity, those positions are not going to be open for recruiting diverse candidates, right? I mean, definitely for Title 42 searches, there has to be, you have to advertise in diverse places. There's a committee that looks at where you're pushing this out to. The committee itself has to be outside just NIAID. It's got to be fairly representative. I mean, usually it's within NIH, but it can also be, depending on the position that you're recruiting for. You can use people from FDA or BARDA, other HHS agencies, if that was, particularly if somebody on the committee had relevant expertise to field candidates for that job. So, that's the way that NIH tries to attract, to give more opportunity to diverse slates of candidates.
We have time for maybe just one or two more burning questions, please.
I like one on this list. What are one of the most impactful moments you've experienced personally in NIAID so far?
No pressure.
I have to say, I guess I sort of, I've been responsible, so if I were gonna go through the whole cycle of coming up with a new idea, securing funding, writing an RFA, publishing it, going through the review process, this is what program officers do. And I was never a program officer, but I did do this in the Division of AIDS for, I mean, you probably have heard of the President's Emergency Plan for AIDS Release, which is a PEPFAR, it's HIV care, prevention, implementation in the global world, in places where the global epidemic is severe and there wasn't very good health care infrastructure for dealing with it. So we had some PEPFAR projects that we secured the funds for. I mean, I saw it through all its steps, and I have to say, the day that we decided upon awards and we knew that they were going out the door and we started meeting with the investigators on what they were going to do, and then I guess you can go years beyond that, knowing that they published something or did something for PEPFAR. Going through that whole cycle made me feel really gratified. So it wasn't a moment, it was years. But that's how science is, right? I mean, the things that we're doing, the things that are happening now in the laboratory, the RO1s that we fund, it's like you have to abstract from what they're doing and think down towards the horizon that's years away for what impact they're going to have. And that's exciting for some people, and if you have to be a little bit of an abstract thinker to see it and get excited over it, or maybe you just have to be really geeky, I don't know. But seeing that whole cycle happen and then, reaching public, seeing that it helped reverse an epidemic, that was gratifying.
Excellent. Thank you so much.